To receive instant notification each time I upload, please hit the subscribe button and the bell notification.
Lockhart Perspective. Helping you along the way. Helping you to make it through another day. Yeah. Everything you need to know that you never knew that you need to know. Lockhart Perspective.
Well, hello, and welcome. We are simulcasting today. We are both on Headlines with a Voice as well as the Lockhart Perspective. And as you can tell by the title, we've got a lot of connecting of the dots to do. Today, we're going to be looking at the actual act of chimeras. We're looking at exactly how they're made, what does it mean to mankind, and I really want to talk about how we get so distracted by other things, information that's old that others may know about and others don't. And so it distracts the masses just in time for them to slip something in. We're going to be looking at a lot of research today. And the purpose for that is because we have to substantiate what it is that we are putting in the forefront. We are definitely looking at what the scientists have done, not only in the study that we have listed down here on the bottom, but we're also going to take a look at some of the breakthrough that has gone on there in Israel. Well, they've come across some really amazing things. I want to say hi to all of you. So let's just go on ahead and look at exactly what we're talking about first. The first thing that we're going to look at is some scientific stuff, right? Yeah, let's look here. I got some information and I want you to look at it. Pretty boring when you just look at it from face value. And you know what? A lot of times I think that a lot of stuff appears to be boring, so that unless you're a nerd like me or somebody unique like I am, you're just not going to look at it. You're like, gee whiz, it's just too much reading. Well, I don't want to go into all of that. Why would I want to look at it? Well, it's okay. I've got you. Don't worry. 40 Boom, I see you have a heart there. Hello, and hello to Daryl Calhoun, King BJ, Mona. Is that Mona? Yeah, that's Mona. Tonette, how are you today? Damn Yankees, daughter. How you doing? All right, John. Good to see you, as well as Starfire and Alan Child. It's good to see all of you today. Now, let's break it on down. Rage Against the Machine. I love you, sweetheart. Thank you for being here. Let's break it down, you guys. I got to break it down because I can't talk about a topic unless I'm willing to go down deep into the dirt to give you the information that you need. Purposefully, these links have not been added to the description prior to the show's performance simply because I don't want anyone to run off and just have this information and not know how to put it in context. You got to be responsible. Now, this is the viable rat mouse chimeras. Where do we go from here? Now, this story was published. This is not an actual story, it's an actual publication of a study that was done. 2010 is where we're going here with this one. Well, we're going to go up. You know me. We got to connect the dots. We got to start as far back as we can, and then we got to come on up. However, we've also got a couple of clips, and some of those clips go back as far as 2009. 1991 that show you that you have been desensitized to the welcoming of chimeras. And there's a major deal that I don't think a lot of people connect with what we're talking about today. And that is that there's another way you've been desensitized. Every time you take your camera on Instagram or TikTok or any other app that has an animal and it's so cute and fuzzy, and then you put it all over the place on your Facebook, you put it on your Instagram, you change it to your logo, and it's you with a little kitten nose or a little dog nose. Believe it or not, those are forms of desensitization. Felicia, how dare you? Oh, well. Let's take a look here. Now, the main text here is experimentally produced chimeras between different mouse strains. All right. Now we have been exceedingly, I want you to look at this first. This is what I want you to do. Let's go back because I looked at this earlier, and I want you guys to go here first. All right. Okay. Just so that we can have it bigger so you can see it. In a tour de force study, Kayabashi et al. 2010 described the first viable rat mouse chimeras and demonstrate that rat induced pluripotent stem cells can rescue organ deficiency in mice. It goes on to say that rat iPS cells formed a fully functional pancreas when injected into the mouse blastocysts, lacking the PDX1 gene required for pancreas formation. Now, the main text goes on to say that experimentally produced chimeras between different mouse strains have been an exceedingly useful tool for the developmental biologists, contributing to our understanding and the establishment of cell lineage, cell determination, and the development of the immune system and other organs. In this issue of Cell, Kobayashi dramatically extended the potential of mammalian chimeras with their report of viable rat mouse chimeras that can develop to term and become fully functional adults. Need you guys to let me know if you can hear me over there well. Yep, I think you can. I just saw that. Yep, you guys are cool. All right. Into fully functional adults. Be with me. This is the boring part, but we're going somewhere. So now in their study, Kobayashi and colleagues relied on previous knowledge, but also added a few new wrinkles. They first derived mouse and rat embryonic stem cells and induced pluripotent stem cells using the standard methods for the mouse and capitalizing on the recent isolation of rat ES and IPS cells. Both mouse and rat were tagged with different fluorescent dyes, allowing the authors to follow their distribution in the developing chimeras. The authors wanted to prove, at least in principle, that exogenic organ complementation. I'm laughing because wait till you see what's after this. Exogenic just threw me. Wait till you see this. Could be achieved. That is, that donor cells of one species could rescue a defect in an organ development in a recipient of a different species. Please follow the key words of a different species. So, as first step, they set out to produce viable chimeras between rats and mice, even though many previous efforts to make such chimeras had failed. The only viable intergeneric chimera is that of a hybrid between the sheep and the goat. All right. Now, to test the possibility of the viable mouse chimeras could be formed, Kobayashi injected fluorescently labeled mouse or rat iPS cells into the rat mouse blastocysts, respectively, and in return, returned them to the blastocyst compatible pseudopregnant females, that is, foster mothers of the same species as the blastocysts. The authors then examined the resulting fetuses, newborns, and adults and found evidence of a substantial contribution of donor stem cells to tissues and organs of the host, despite a big contribution of donor cells. The size of the newborn and the adult chimeras was determined by the species of the host blastocysts. It is not clear whether it is the embryo itself or the uterine environment that determines the extent of chimera growth. To distinguish between these two possibilities, one would have to transfer chimeric embryos into the uterus of the pseudopregnant females of the same species as the donor stem cells, not the blastocysts. Previous studies suggest that such experiments would fail because of the need for compatibility between the fetal part of the placenta and the uterus. Here in figure 1 is the actual mouse to rat change, and the mouse-like chimera and the rat-like chimera. And then you see here the rat iPS cells, the host of the host blastocysts. Here is the normal one, and here is the PDX1. Now, that sounds like a lot of stuff, Felicia. So much tongue twisting. Yeah, so much tongue twisting that even me, it's hard to get it all together. But is it really why it's a little difficult? You never know. I want you guys to look at something. That was in 2010, right? Well, this next story is dated December 2020. Now, I'm going to share the screen with you on this one. And I'm tickled pink because wait till you just hallelujah. You know, it's amazing when we see mankind attempt to be Yah, can never ever be done. It can never ever be done. So whether you call him God, whether you call him Yahweh Shah, whatever you call him, I'm saying they could never be Yah, no matter how man tries. This right here is the U.S. University grafts the scalps from newborn, from aborted babies onto humanized mice. Your tax dollars at work? Let's see. December 17th, 2020, the University of Pittsburgh is conducting medical research by grafting scalps from aborted babies onto rodents after publishing a report in the National Catholic Register about the many ways that aborted children are being used for scientific research at a major universities and hospitals in the United States. Stacy A. Traskanos, sorry if I badgered your name, sister girl, tweeted a summary of her horrifying findings. She links the Magellan research to the U.S. COVID-19 guru, Dr. Anthony Fauci. Aborted baby scalps grafted onto mice, their organs transplanted, humanized mice developed at the University of Pittsburgh, all to study the immune system, supported by Dr. Fauci's NIAID, paid for by you and me. Now, this individual is an executive director of the Bishop Joseph Strickland's, the St. Philip's Institute of, I don't know how to say that because I'm not even going to try it, but it's supposed to be something dealing with catechism, with Catholic, and the evangelization in the Diocese of Tyler, Texas. Bishop Strickland retweeted her report, declaring, "These are crimes against humanity that should be prosecuted. Silence in the face of those atrocities simply adds to the evil. Wake up, America. Demand that this stops now." That's what she said right here in a tweet. And along with her tweet, she gives some graphic illustrations about what she's referring to. So let me see if I can let you see this a little bit bigger. So, abortion rights, right? So, this is what they were talking about, deletion rights. And this is why they have so many reasons for you not to get deep into that conversation. Well, because it's a very lucrative business. So here it is. This is the number one. Okay. Number A, then you've got letter A, letter B, and letter C. This was 10 weeks. This was here. A mouse, 0 weeks, 2 weeks, 4 weeks, 10 weeks. And then you have the human skin appendage hair here. And then you have the humanized right in here. All right. And this is the bottom line. And how aborted children are used in the medical research in 2020, Traskanos explained that while currently there is heightened interest in the use of aborted fetal cell lines in the C19 vaccine research and production, the use of electively aborted children by the university, government, and industrial scientists for research is shockingly commonplace. In a scientific report published at Nature in September, the University of Pittsburgh research scientists explain self-consciously the basis of their study. Quote, "Human skin is a significant barrier for protection against pathogen transmission. Rodent models used to investigate human-specific pathogens that target the skin are generated by introducing human skin grafts into immunocompromised rodent strains." The researchers write that, quote, "Human fetal tissues were obtained by the Health Science Tissue Bank at the University of Pittsburgh. Human fetal tissues for the constructing humanized rodents were handled and processed under biosafety level 2 conditions." Tranzacos uncovered that the full thickness fetal skin was taken from humans aborted at the gestational age of 18 to 20 weeks of pregnancy at the McGee Women's Hospital and the University of Pittsburgh Health Sciences Tissue Bank. Full thickness human skin from fetuses was grafted onto rodents while simultaneously co-engrafting the same fetus's lymphoid tissues and the hemopoietic, I'm sorry if I mispronounced that, stem cells from the liver, so that the rodent models were humanized with organs and skin from the same child, this particular doctor wrote. Hmm. Well, Felicia, that's nothing. That's absolutely nothing. What else do you got? Because we can take it. Well, let's get a little closer, shall we? Let's get a little closer to today. Let me go ahead and give this to you too. So you can just have it for yourself, just for cracks and giggles. You just may want it to, I don't know, share with people. Let me make sure that you can see it on this side. You can. I'm going to go ahead and give you the entire screen for this one. All right. There you are, entire screen. Now, this was just published. Scientists transplant human brain cells into the brains of baby rats. Scientists have transplanted human brain cells into the brains of baby rats, where the cells grew and formed connections, according to new research. It's a part of an effort to better study human brain development and diseases affecting the most complex of organs, which makes us who we are, but has long been shrouded in mystery. Quote, many disorders such as autism and schizophrenia are likely uniquely human, but the human brain certainly has been very accessible, said Dr. Sergei Pasca, the senior author of a study describing the work published Wednesday in the journal Nature. Goes on to say approaches that don't involve taking tissue out of the human brain are, quote, promising avenues in trying to tackle these conditions. Here is the brain organoid rats. Now, you're not able to see this. There's a little thing that lets you know that this is a photo. But if I go back to the regular one, I'm probably going to lose it and won't be able to get back here. But there's an actual picture of it. So it says here, this microscopic image provided by Pasca's lab at Stanford Medicine shows a human astrocyte cell center in the yellow and the human glial cells scattered in blue inside the brain of a rat. And it's been furnished by the AP. Now, the research builds upon the team's previous work creating brain organoids, tiny structures resembling human organs that have also been made to represent others, such as livers, kidneys, prostates, or key parts of them. To make the brain organoids, Stanford University scientists transformed human skin cells into stem cells and then coaxed them to become several types of brain cells. Those cells then multiplied to form organoids resembling the cerebral cortex, the human's brain's outermost layer, which plays a key role in things like memory, thinking, learning, reasoning, and emotions. Scientists transplanted those organoids into rat pups, 2 to 3 days old, a stage when the brain connections are still forming. The organoids grew so that they eventually occupied a third of the hemisphere of the rat's brain where they were implanted. Neurons from the organoids formed working connections with circuits in the brains. In the brain, human neurons have been transplanted into rodents before, but generally adult animals, usually mice. Pasca, a psychiatry professor at the Stanford School of Medicine, said, this is the first time these organoids have been placed into early rat brains, creating the, quote, most advanced human brain circuitry ever built from human skin cells, and a demonstration that implanted human neurons can influence an animal's behavior, closed quote. To examine a practical use of this approach, scientists transplanted organoids into both sides of a rat's brain, one generated from a healthy person's cells and another from the cells of a person with Timothy syndrome, a rare genetic condition associated with heart problems and autism spectrum disorder. 5 to 6 months later, they saw effects of the disease related to the activity of the neurons. There were differences in the two sides' electrical activity, and the neurons from the persons with the Timothy syndrome were much smaller and didn't sprout as many extensions that pick up input from nearby neurons. The team of researchers whose study was funded partly by the National Institutes of Health, that means you, that means me. Well, they said that they could do the same sorts of experiments using organoids made from the cells of people with disorders such as autism or schizophrenia. Now, let me come over here for a really quick second. Just a really, really quick second, because I want to add something and I want to share with you how I think about things. Here's how I think about things. Whenever you hear me say there's something to this part of California's story where they're saying now they want to just scoop up all these people who are mentally ill or quote unquote mentally ill, and we don't know where they're going to go because, well, listen, in California right now, they don't have any place to put the standard unhoused, I should say. So where are we going to put all the unhoused people who have health challenges? Do you remember that study? We talked about that the other day. There's a story we did just a few days ago, about a week, where California says that if you have people that you know of, anybody can literally go to a judge and say, hey, I think this person's having a bit of problems. They may be schizophrenic, they may be something, but they picked on the homelessness to make everybody say, yeah, it's a good thing that we get them some help. Sure. But then if you can't find them, what do? If you don't know where they are in 2 weeks, what do? If you haven't been able to find them in a month and you cannot locate them because, again, there is no place to put the unhoused standard people, what do? Hey, don't throw rocks. I'm just asking a question. Let's continue. So the story goes on to say that the team of researchers whose study was funded partly by the National Institutes of Health, which is you and me, again, I said it, said that they could do the same sorts of experiments using organoids made from the cells of people with the disorders such as autism and schizophrenia, keyword, in the story that we talked about last week, and potentially learn new things about how these conditions affect the brain, too. Dr. Flora Varcarino of Yale University, who previously grew lumps containing cerebral cortex that were made with DNA from people with autism. That was the study that this person did. Said the study moves the field forward. Quote, it's extremely impressive what they do here in terms of what these cells actually show us in terms of their advanced development in the rat, said Varcarino, who wasn't involved with the study. Such experiments in animals raise ethical concerns. For example, Pasca said that he and his team are cognizant of the rats' well being and whether they still behave normally with the organoids inside them, which he says they do. Still, Pasca does not believe that this should be tried in primates. Well, you also have the ethicists also wondering about the possibility of brain organoids in the future attaining something like human consciousness, which experts say is extremely unlikely now. Really? Well, you already know I got something, right? Some scientists are studying human brain organoids outside of animals. For example, the researchers at the ETH Zurich in Switzerland published a study in Nature earlier this month at the time of this publication, describing how they are growing brain-like tissue from stem cells in labs and then mapping the cell types in various brain regions and genes regulating their development. Some are using these structures to study autism. Pasca said brain organoids could also be used to test the treatments for neuropsychiatry disorders, the largest cause of disability worldwide. Such research, he said, should help scientists make strides that have been extremely difficult until now because it's hard. It's so hard to get at the human brain, he says. He said that it is the reason why we're so much more behind in psychiatry compared to any other branch of medicine in terms of therapeutics. Well, that's what he said. Now, here is what I want to tell you, that there has been a lot more research than a lot of people think. These two studies and the stories that we just got through looking at, they were nowhere as close to today as the next one is. The next one is very close to right now. And I'm going to share that with you because this one tells you a lot about exactly where we're headed and how far, while all of us were sleeping, we've gotten. This is the scientist's report creating the first embryo with human and non human primate cells. Now, this was some groundbreaking information that came out of Israel. That's right, Israel. Hmm. In a groundbreaking experiment, researchers have successfully created the first human monkey chimera. The work published in the journal Cell describes the first embryo containing both human and monkey cells that was cultured for 20 days, led by Juan Belmont. I'm not going to do the whole Carlos and the other name because I don't want to badger it. But nonetheless, it does go on to say that the study represents the culmination of decades of work in understanding early embryo development in a non-human species, which Belmont hopes will now apply to humans, but it is bound to raise serious ethical questions about the implications of combining human cells with those from a different species. Let that just sink in, even if it is a closely related one. And the report was accompanied by commentary from ethicists on how the work should be interpreted and what the careful next steps should be in pursuing this line of study. Belmont, a professor in the Gene Expression Laboratory at the Salk Institute of Biological Studies and well respected for his work in the embryo development, is very clear about why he pursued the experiment and where he hopes that it will lead, creating cross species mutants to capitalize on specific physical features or characteristics, X-Men style, was most definitely not the goal. In interviews with Time on the experiment as it progressed since 2019, he carefully laid out the biological mysteries he hoped to solve and the gaps in knowledge about early development he wanted to fill. The Iranian Supremian leader is another story that you guys can pull up here. If you get to this one, you click this one, and you're going to be able to go down and see more stuff once you get this link. So you'll be able to entertain yourself for quite some time. What I want to do right now is I want to take you guys on a little bitty look of some of the things that happened when the scientists decided that they wanted to soften you up just a smidge. It's so important to get you on their side. Don't you understand? Don't you know? Because if you just listen to the ramblings of a 58-year-old woman like myself, you'll possibly think that I just don't want you to have any fun. So, what we're going to do right now is we're going to take a look. We're going to take a look at this. This is something that we talked about a little bit over a year ago, and I want to bring it to your remembrance because it's very important. This is what they did. In tandem with you having all of these apps that you can make your face look like a little lion or a little bear or a little soft, cuddly kitten. This is the movie that they gave you. It was a small mini series. It didn't go very far, but here it is. It's called Sweet Tooth. This is my commentary on that a little while ago. Let's check it out. Sweet Tooth. It's a Netflix series. And this Netflix series still had my grandkids
He's
in the
happy
background.
that he's got his brother a drink. All right. So, loud as it may be. On a perilous adventure across the post-apocalyptic world, a lovable boy who's half human and half deer searches for a new beginning with a gruff protector. So that's the gruff protector in the background. And it stars Nunso Anozi and Christian Covey, as well as Adil Akhtar. These are some of the pictures that you will see. This little boy has antlers. This is that cute little filter that I kept telling you about, that I was just talking about. That's the filter that I was mentioning for so long. I kept saying, why are all of these things with these filters? It just didn't make sense to me. But now we see. So transhumanism is something that is really on the rise right now. And while we're looking at these things, we really need to ask ourselves, why? Why were they so giddy about everybody and their mama having a filter that would make them look like a little puppy or an animal? And if you notice, they always make it look like the most genteel. When we all know that a lot of the real transhuman things in the past from all different times have not been very genteel at all. So I just thought that I'd come and share this with you because when I saw this, I said, there's the answer to the question why. There is the answer to the question why. So again, it is on Netflix. And this is one of the trailers. You can see it. It's a trailer. I'm not sure if there's a trailer that we could see that would be, without cost on so many levels. But if you want to, you can definitely go and check it out. I'm going to take this link and I'm going to place it down below inside of the comment box so you all can see it. Yeah, I think it's creepy too. I really do. So there it is. That'll get you there. But therein lies the answer, because I had been asking for so long. I said,
Forced organ harvesting from Falun Gong practitioners and other political prisoners in China has raised increasing concern within the international community. According to a report by former lawmaker David Kilgore, human rights lawyer David Matas, forced organ harvesting from Falun Gong practitioners and other political prisoners in China has raised increasing concern within the international community. According to a report by former lawmaker David Kilgore, human rights lawyer David Matas, and journalist Ethan Gutman, political prisoners, mainly Falun Gong practitioners, are being executed on demand in order to provide organs for transplant to recipients. The organ harvesting has taken place both as a result of the Chinese Communist Party's persecution of Falun Gong and because of the financial incentives available to institutions and individuals involved in the trade. Now, China has been investing heavily in controversial research on using genetically modified pigs as a source of human organ transplantation, touting it as a world first achievement. Now, I know if you're like me, you're asking, how could they do this? Well, xenotransplantation. It's a medical procedure whereby animal organs are transplanted into humans. And according to a Xinhua report that was published September of 2020, gene editing related startups in China alone had received the equivalent of $310 million in funding. Get this, I want you to hold on to this really, really carefully because this is important. Luhan Yang is the founder of Huihan Biotech in Hangzhou, China. She's also the co-founder and chief scientist of the Cambridge gene editing company eGenesis in Boston, Massachusetts. She and a team of scientists have created a prototype xenograft with clinical potential, successfully solving two of the major xenograft safety challenges. Now, the first one was removing porcine endogenous retroviruses from pigs and enhance xenograft immunocompatibility. Now, Yang's research was also published in Nature Biomedical Engineering in September of 2020. The co-authors of the paper include, get this, Huihan Biotech, Harvard University, Massachusetts General, eGenesis in Boston, Massachusetts, and Yunnan Agricultural University in China. The paper describes how CRISPR-Cas9 knockout and transposon gene insertions technologies were used to modify a group of genetic cassettes in pigs to further enhance the pig's immunological compatibility and blood coagulation compatibility with humans. The paper says, quote, the engineered pigs exhibit normal physiology, fertility, and germline transmission of the 13 genes and the 42 alleles edited, close quote. Prior to this, there had been several technological breakthroughs in genetically modifying pigs to make organs more suitable for human transplantation, several of which resulted from the work of Yang's mentor at Harvard Medical School, Professor George Church. Now, as a little side note, something that I thought was unique,
Church has been an outspoken individual as it relates to following a vegan lifestyle for reasons concerned with health and environmental and moral issues. But I just thought that that was a bit unique, that this is something that he believes in, not eating animals. But in 2015, Yang, along with Church, founded eGenesis in Boston, which received $38 million dollars and $100 million dollars in two rounds of funding in 2017 and in 2019, and another $125 million dollars in March of 2021, and a third investment round. There's somebody pouring a lot of money to figure out how to make chimera, human chimera. 1 Corinthians 15:39 says, all flesh is not the same flesh, but there is one flesh of man, and another flesh of beasts, and another of the fish and of the birds. So no matter what you do, you could never be the Most High Yah. Remember, we've been here before. This has been tried time and time again. And the last time it was water. The next time, it'll be fire. This is Felicia Lockhart, and this has been the Lockhart Perspective.
So that is something that we did last year. Both of those two little videos that we had done. However, we have a whole lot more inside of our playlist on the Lockhart Perspective, dealing with hybrids, dealing with things that are like the the liger, the tiger mixed with something else. So here's the deal. You got to try this stuff out on the animals first before you can then roll it out and try it on humans. I guarantee you that even though we're seeing a lot of individuals within these studies say, we have to wait because we don't know about the ethics piece. We don't know about this or that. I completely believe that while you're doing all that study, you're already pushing past and making sure that there are laws put into place that will sort of circumvent your issue. You won't have a problem. They'll roll it right out to you. They'll roll it right out to you. Listen, you had people lining up in droves to give away their DNA just recently as a sport, something that was done specifically for entertainment purposes. Well, it said it on the insert, and that was to give up their DNA just to find out where they came from. The problem is they never told you that they were going to do whatever they wanted to do. Well, let me correct this. They told you, but you didn't read it. Anton Daniels, a very good friend of the channel, made a mention of that today while we were chatting about some other things on his channel. And he was saying, hey, you know what? Some people don't even read what's inside for their cell phone agreements or what's, for this app or that app. And I'm paraphrasing here. Okay. But he mentioned that today. And he's absolutely right. That a lot of us have information that's right there at our fingertips. We never know what we're signing on. And yet people in droves signed up to give up their DNA and don't even know what type of study it was done. That was done on it. None of them. Now, there's other things, of course, as we look at the whole Roe thing, the Roe situation that happened a couple of months ago where they went out and said, oh, well, somebody just leaked this information and somebody just did this or that. And it was not a leaked information just because, well, listen, we've had the Supreme Court for a bazillion years. And all of a sudden, something as pivotal, something as detrimental, something as polarizing as this particular topic was leaked. Have we ever gotten to the end of that? I don't think so. But you will definitely know that there had been legislation in different states that was prompting people to do whatever they could do. And we just got through seeing the studies that proved that they are able to do a lot with the remains that you choose not to have. In tandem with that, we're looking at all of the little children run around with the little noses, grown women changing their icons on their pages to have the little kitten face, and nobody is understanding. So let me explain this to you. And where am I coming from? Where I'm coming from is the racism that we speak of. Letter A is going to be dealing with all of the robots and the humanoids that they're creating, ones that actually have human skin. Please don't believe me. Just look it up. And then the second thing is going to be all of these chimeras. Well, you're not supposed to dislike them. You're not supposed to talk about them. Well, because they're human. Well, they have a specific level of DNA in them. And this makes them human, Felicia. And there's still some people after listening to this would say, oh, I'll never buy any of that. You are being desensitized, my brother and my sister, every single time you happen to look at one of these types of shows. And here's one I want to show you from years ago. And they're going all over the United States and all over the world. China is notorious for doing this type of research. Here was a show that they had several years ago. I want to show you this one. It's called the Chimera TV series. Now, this was it here. And it was a journalist that investigated. Let me make sure that you can see it, because oftentimes I'll be just chatting away, just chatty Cathy, and you guys can't see nothing I'm talking about. A journalist investigates the death of his girlfriend at a fertility clinic where she worked and uncovers a plot to create a new breed of human based on crossing the genetics of a man and an ape, Chimera. This was back in 1991. Back in 1991. And here's another one. Let me give you this one. It's another television series, but this one was overseas, and I believe it was 2009, was it? Let me see. 2009 or something. I'm just going to give you the clips because it doesn't matter when it was. The point I'm making is that for a long time, they've been smoothing things over with you. They've been softly letting you see exactly what they mean. See that? Let me read it to you really quickly before we close. So this is a show called Chimera. It's in 2021. You see that? 2021, the synopsis. 35 years ago, the nation was rocked by a series of explosions that claimed the lives of numerous victims, set off by a serial killer with a sick and twisted agenda. The string of horribly violent crimes became known as the Chimera case. After leaving a trail of chaos and carnage in his wake, the Chimera killer eventually disappeared. Once you get into the story, you'll see even more. The point that we're making today, ladies and gentlemen, is that we've been programmed. Every single one of us have been programmed. Whether we know it or not, we have been programmed. But here is the good news. You don't have to be. Take the information, look at it. Don't be afraid of this stuff. Don't let any of this stuff make you lose one night's sleep at all. It's information. It's just information for you to understand what to accept and not to accept. That's it. That's all it is. It's nothing for us to go around like this all day long and all nervous and all schizophrenic about it. We're just, I got this mindset. Now I got this mindset. No, I think about this mindset. No, I've got this problem. We don't need to go there. All we need to do is be equipped with the knowledge that we have and be equipped with the word. Be equipped. That's all. And you need to know that as much as you see all of this coming up, it only means it's time to go. It only means that it's time to charge in advance toward what the word has told all of us. I appreciate each and every one of you guys, and I thank you for giving me the opportunity to come before you today and share some of this information with you. I believe that whenever we're starting to look at a whole lot of stuff that's going around that has absolutely no significance in our personal lives, we need to look a little deeper, and we'll find that there is some information that could help us make better decisions about whether or not we go and give up plasma, or about whether or not we go and we do X, Y, or Z with our body. There's a lot of information to be had, a lot. And there's a lot to be said about a person who actually studies to find out exactly what's really going on. As I always say, what's it all about, Alfie? Before we leave, I want to hug on Saman's neck here. And there is the ability for you guys to give down there at the Cash App. It is Lockhart Perspective, and you can give at your discretion. You can give a buck. You can give whatever you want to give. You can also give here on the channel, should you so elect to. We also have a PayPal account. If you'd like to give there, we'll gladly receive that as well. We're going to go on ahead here and let me see some folks here. Who do we have? Thank you very much. Zenren, Arizona, AZ, thank you so very much for supporting the channel. We definitely appreciate that. Thank you, thank you, thank you. And let's see, who else do we have here that's with us today, Zenwen? Who else is visiting us? Well, we have Walk in Truth. Of course, we have Pam. How are you today? Donna, good to see you. Again, Tonette, hello. Is that Shart? Schnart? How do you say that? You guys know how I get with pronouncing things. I don't want to do it incorrectly, but I want to say hello to you as well. Oh, Delane, how are you doing? Good to see you. Of course, 1381, how are you? Be Borrowed, good to see you. Jane Doe, hi. Lady Homestead, it's good to see you as well. Who would, Old Head Deuce, get out of here. How are you doing? Well, aren't you a blast from the past? It's good to see you. And Sandra Cummings, good to see you as well. Well, it looks like someone just blessed us again, and that was Sharon Cardamone. Hello, how are you, Sharon? And I so appreciate you for doing that and thinking of us here at the Lockhart Perspective and Headlines with a Voice. Thank you guys so very much. William Smith, I see you. Betty R., it's good to see you as well. My sister, Black Spring with Autumn, thank you so much for being here. Is it Spike in Madness? Hello, salutations to you as well. Salutations to you as well. Good Shepherd, how are you doing today? It's good to see you. I like that. The Good Shepherd. That's good stuff. Good Shepherd. Yes, he is. It's great to see each and every one of you, even those who have not had the opportunity to come inside the chat. I want to thank you for supporting us here. You guys are really, really the best of the best, and you make us who we are here at the Lockhart Perspective, as well as the Felicia Lockhart Group. Jane Doe says, imagine the mentality of servicemen going to a donkey show or being a willing participant that made nerves raw. Can't you see choices difference? Yes, I hear you loud and clear. I hear you loud and clear. You guys, I want to tell you something. There's a lot of stuff that's going on right now, a whole boatload of stuff. Lady Homestead says, thank you for the information. You are so very welcome. I'm going to put all these links down as soon as we're done. You guys will be able to glean them and pull them at your discretion so that way you could have them. But there's a lot more stuff. As you guys know, if you guys subscribe to Nature, if you guys subscribe to any of the science papers, you'll be able to find a lot of stuff the first few days that it hits. But the best part about it is that we were actually able to pull together an entire storyline or matriculation from one year to the next of how this thing grew. KRWN, they also talk about the mixing of genes. Here we are. The mixing of genes of different species and Morbus on Netflix. Okay, I'm going to have to check that out. Thank you for being here, KRWN. I want to take this opportunity to thank everybody who has come over here from anybody else's channel, whether it's been because of Anton Daniels' channel, whether it's become the Millionaire Morning Show, whether it's been because of Zara B, whether it's because of Jay Prince. I want to thank each and every one of you that helped us today make it to number 4,000. I hope that's what it is, because that's what it showed earlier today. But we have finally, after all of these years of chugging away and chugging away, having dynamic stories like some of the things you saw today get only 100 views, get only 200 views, and we would, Ronnie.d.Mann and I would get highly excited when we would see 100 views. Some videos that took me about a month sometimes to compile, getting all the pieces together, and then you only see 3 minutes of it. I only get 100 views on it, but I want to tell you guys something. You guys are what makes the difference. You helped me get to 4,000 subscribers, and I want to make sure that you know from the bottom of my heart that I appreciate you, and I appreciate you for rocking out with us over here. So no matter how you found this channel, no matter where you came from the bottom of my heart, baby, I want to tell you thank you. And thank you, Sister Black Spring, for taking care of us. Thank you, Precious. We so appreciate you. Now, if you've not gone to Black Spring's channel, please do. She's got a great show over there, and a great channel. She does really good work. Oh, you found me from Lisa R. Hall and Marfoogle News. I love Marfoogle News. I love Marfoogle. He is hilarious, and so is Lisa. Lisa is my she and I, we're the twins. We are the tinfoil hat fedora sisters. Mine is red and hers is blue. We both chose the colors that we like. Drops Family Garden. Hey, it's good to see you. It is so good to see you. And Donna Moss says, I appreciate the truth. Well, we appreciate you for being here, y'all. Hey, Margaret, how's it going? I appreciate you. I'm new, honey, and I'm glad you are. My mods are the best. This chat is the best. They will love on you. Some days, if I can't see you and I've not said anything, no worries. They got you. They're going to spread all the love on you. Listen, you guys, I appreciate each and every one of you for being here. Do me a big favor and remember what we always say on any Felicia Lockhart Group publications, and that is that man shall not live by bread alone, but by every word that proceeds out of the mouth of the Most High. Until we see each other again, I want you to make sure that you take care of yourself first. I always tell you that if we don't take care of number one first, honey, let me just let you know, you ain't going to be no more good. Not no more good at all. And so I want to remind you to take care of yourself first and then reach around, pull somebody else up and help them as well. Those that you love, those that are in your family, somebody in need, you got enough stuff in you, baby, to go ahead and take care of them. Until you and I see one another again, I want you to continue, please continue to be blessed. Y'all take care. To receive instant notification each time I upload, please hit the subscribe button and the bell notification.